The evolution of microbial life. Our planet's history began about 4.6 billion years ago. 3.5 billion years ago, the first living organisms appeared. The earliest organisms were all prokaryotic, meaning these were organisms that were single-celled and they don't have membrane-bound organelles or membrane-bound nuclei. The early atmosphere lacked oxygen. 2.7 billion years ago, atmospheric oxygen began to accumulate as a byproduct of the photosynthesis of autotrophic prokaryotes. The first eukaryotes appeared about 1.8 billion years ago. Eukaryotes are composed of one or more cells, and they contain nuclei and other membrane-bound organelles. The first eukaryotic organisms to appear on Earth were the protists. Protists are mostly microscopic and unicellular, and they are represented today by a great diversity of different species. Eukaryotic cells most likely evolved from ancestral host cells that engulfed smaller prokaryotes. This idea is called the endosymbiotic theory. The mitochondria of our cells, and those of every other eukaryote, are descendants of those prokaryotes, as are the chloroplasts of plants and algae. The oldest fossils that clearly showed multicellular organisms are about 1.2 billion years old. The first multicellular organisms were simple and very small. Large, diverse multicellular organisms did not appear in the fossil record until about 635 million years ago. The Cambrian explosion began about 541 million years ago and marked the beginning of the Paleozoic era. In the Paleozoic era, about 500 million years ago, the dry land on Earth began to be colonized by plants, fungi, and insects. The Mesozoic era is sometimes called the Age of the Dinosaurs. During the Mesozoic era, more and more familiar organisms begin to dominate the landscape, including flowering plants, birds, and mammals. Modern humans are Homo sapiens, and we appeared roughly 195,000 years ago. Early in its 4.6 billion year history, our planet had no lakes, no oceans, and very little oxygen. At this time, Earth was bombarded by debris that generated heat that vaporized all available water. About 4 billion years ago, the water vapor had condensed into oceans on the planet and the surface cooled. Volcanic eruptions continued, adding carbon dioxide, methane, ammonia, and other nitrogen compounds into the atmosphere. One of the more popular hypotheses for the origin of life is called the four-stage hypothesis. It is believed that the first organisms were products of chemical evolution in 4 stages. The first stage involves the synthesis of small organic molecules, such as amino acids and nucleotide monomers. The second stage involves joining of these small molecules into macromolecules, including proteins and nucleic acids. The third stage is the packaging of all these molecules into pre-cells, which are droplets with membranes that maintained an internal chemistry different from its surroundings. The fourth and final stage is the origin of self-replicating molecules that eventually made inheritance of genetic material possible. Let's look at each of these stages more closely. In stage 1, we have the synthesis of organic compounds. The chemicals that formed on Earth just after its formation included water, methane, ammonia, and other small inorganic molecules. However, the complex organic molecules needed for life, including sugars, fatty acids, amino acids, and nucleotides, are composed of these same elements. But the question remained, could these complex molecules have arisen from these small inorganic molecules? In 1953, Miller and Urey performed an experiment. They created an apparatus to simulate the conditions of the early atmosphere of Earth. They included an atmosphere containing hydrogen gas, methane, ammonia, and water vapor. They included a flask of warmed water to simulate the primordial sea. After about 1 week, the apparatus contained an abundance of organic molecules essential for life, including amino acids, which are the monomers of proteins. These molecules had shown up in the water of the apparatus that was representing the primordial sea. There are a lot of different theories about where life began. These theories include submerged volcanoes, deep-sea hydrothermal vents, and meteorites. Meteorites contain some organic molecules, including amino acids, lipids, simple sugars, and even nitrogenous bases. Stage 2. The abiotic synthesis of polymers. Once small organic molecules were present on Earth, how is it possible that they were linked together to form polymers such as proteins and nucleic acids without the help of enzymes or other cellular equipment? It has been demonstrated in the laboratory that such polymerization can be brought about by dripping solutions of organic monomers onto hot sand, clay, or rock. The heat vaporizes the water in the solution and concentrates the monomers on the underlying material. Some of these monomers then became spontaneously bound to one another, forming polymers. On early Earth, raindrops or waves may have done the same thing, concentrating organic monomers onto fresh lava or other hot rocks, allowing polymers to form. Stage 3. The formation of pre-cells. A key step in the origin of life would have been the isolation of a collection of organic molecules within a membrane. These molecular aggregates are called pre-cells. Researchers have demonstrated that pre-cells could have formed spontaneously from fatty acids. The first membranes were likely quite porous, allowing organic monomers, such as RNA nucleotides and amino acids, to cross freely. However, polymers that formed inside these pre-cells were too large to exit. They may have had the ability to use chemical energy and grow. Pre-cells made in laboratory experiments have been shown to divide, producing new pre-cells in a simple form of reproduction. Stage 4. The origin of self-replicating molecules. What were the first genes like? One hypothesis is that they were short strands of RNA that replicated without the assistance of protein enzymes. Laboratory experiments have shown that short RNA molecules can assemble spontaneously from nucleotide monomers. When RNA is added to a solution containing a supply of RNA monomers, new RNA molecules assemble that are complementary to the parts of the starting RNA. Some RNA monomers spontaneously join, forming the first small genes. Then, an RNA chain complementary to one of those genes assembles. If the new chain, in turn, serves as a template for another round of RNA assembly, the result is a replica of the original gene. Each pre-cell would contain its own population of RNA molecules, each with a random sequence of monomers. Some of these molecules would self-replicate, but their success at this reproduction would vary. These pre-cells that contain genetic information that helped them reproduce more efficiently than others would have increased in number, passing their abilities on to later generations. Mutations would have resulted in additional variations on which natural selection could work. The most successful of these pre-cells would have continued to survive, reproduce, and to evolve. At some point during millions of years of selection, DNA, which is a more stable molecule, replaced RNA as the storehouse of genetic information. These protocells passed a fuzzy border to become true cells. The human microbiota is the community of microorganisms that live in and on our bodies. Some of our intestinal inhabitants supply us with essential vitamins. They also enable us to extract nutrition from food molecules that we would not be able to digest otherwise. Many of the bacteria that live on our skin perform helpful housekeeping functions, such as decomposing dead skin cells. Prokaryotes also guard the body against disease-causing intruders. Prokaryotes living in the soil and at the bottom of lakes, rivers, and oceans help to decompose dead organisms and other waste materials. This recycles vital chemical elements such as nitrogen and places it back into the environment. Without prokaryotes, the cycles that sustain life would come to a halt, and all forms of eukaryotic life would no longer be possible. In contrast, prokaryotic life would undoubtedly persist in the absence of eukaryotes, as it did for 2 billion years on early Earth. Prokaryotic organisms come in many different forms. Determining cell shape by microscopic examination is an important step in identifying a prokaryote. The three most common shapes are spherical, rod-shaped, and spiral-shaped prokaryotes. Spherical prokaryotic cells are called cocci or coccus for singular. Rod-shaped prokaryotic organisms are called bacilli or bacillus for the singular form. And spiral-shaped prokaryotes include the spirochetes, which include different species that cause syphilis and Lyme disease. Although all prokaryotes are unicellular, the cells of some species usually exist as groups of 2 or more cells. Cocci that occur in clusters are called staphylococci. Cocci that occur in chains are called streptococci, including the bacterium that causes strep throat. Some prokaryotes grow branching chains of cells. Some species even exhibit a simple division of labor among specialized types of cells. There are even some giant prokaryotic cells that actually dwarf most eukaryotic cells. The giant bacterium, which lives in the gut of surgeonfish, is large enough to be seen with the naked eye and can grow up to 10 times the size of a typical eukaryotic cell. In many natural environments, prokaryotes attach to surfaces in a highly organized colony called a biofilm. A biofilm may consist of one or more several species of prokaryotes, and it may also include protists and fungi as well. As a biofilm becomes larger and more complex, it develops into a community of microbes. The cells within this community communicate using chemical signals, and the members of the community engage in a division of labor, including defense against invaders, among other activities. Biofilms can form on almost any type of surface, including rocks, organic material including living tissue, metal, and plastics. Biofilms are common among bacteria that cause disease in humans. A biofilm known as dental plaque can cause tooth decay. Ear infections and urinary tract infections are often the result of biofilm-forming bacteria. The structure of biofilms makes these infections especially difficult to defeat. Antibiotics may not be able to penetrate beyond the outer layer of cells, leaving much of the inner community intact. Binary fission is the process by which most prokaryotic cells divide. Some species can produce a new generation in just 20 minutes under optimal conditions. If reproduction continued unchecked at this rate, a single prokaryote could give rise to an entire colony that outweighs the Earth in only 3 days. Each time DNA is replicated prior to binary fission, spontaneous mutations are able to occur. Such mutations can generate genetic variations in prokaryotic populations. If the environment changes, an individual that possesses a beneficial gene can quickly take advantage of new conditions. So, why are we not knee-deep in prokaryotic organisms? Well, several factors limit prokaryotic cell division, including limited resources such as food and space, a buildup of metabolic waste products that eventually pollutes the colony's own environment. Some prokaryotes can survive during very harsh conditions by forming specialized cells called endospores. An endospore is a thick-coated protective sphere produced within the prokaryotic cell when the prokaryote is exposed to unfavorable conditions. With the endospore, the prokaryote then can survive such things as trauma and extreme temperatures. When the environment becomes more hospitable, the endospore can absorb water and resume growth. The prokaryotic pathways that transform energy and matter are far more diverse than those in eukaryotes. Some species harvest energy from inorganic substances such as ammonia and hydrogen sulfide. Soil bacteria that obtains energy from inorganic nitrogen compounds are essential to the chemical cycle that makes nitrogen available to plants. Some bacteria use sulfur compounds as a source of energy and support diverse animal communities. Prokaryotes can thrive in conditions that seem totally inhospitable to life. Prokaryotes have been found between layers of rock buried hundreds of feet below the Earth's surface. They've also been found near hydrothermal vents where scalding water and hot gases surge into the sea more than a mile below the surface. Some prokaryotes are considered symbiotic. Symbiosis is a close association between organisms of two or more species. For example, many of the animals that inhabit hydrothermal vent communities harbor sulfur bacteria within their bodies. The animals absorb sulfur compounds from the water, and the bacteria uses these compounds as an energy source to convert it into organic molecules that in turn provide food for their hosts. Nitrogen fixation is the process of converting atmospheric nitrogen into a form usable by plants. Symbiosis with cyanobacteria gives plants an advantage in nitrogen-poor environments. Other nitrogen-fixing bacteria live symbiotically in root nodules of plants in the legume family. Life depends on the recycling of chemical elements between biological and physical components of the ecosystem. Nearly all the nitrogen that plants use to make proteins and nucleic acids comes from prokaryotic metabolism in the soil. In turn, animals get their nitrogen compounds from the plants. Prokaryotes decompose organic matter and return vital elements to the environment that can then be used by other organisms. If it were not for such decomposers, carbon, nitrogen, and other elements essential to life would become locked in the organic molecules of corpses and waste products. Bioremediation is the use of organisms to remove pollutants from water, air, and soil. Prokaryotic decomposers can be used to treat sewage. These microbes decompose the organic matter in the sludge, converting it to material that can be used as landfill or fertilizer. Aerobic prokaryotes and fungi remove most of the organic matter from the sewage. The outflow is then sterilized and released back into the environment. Bioremediation has also become an important tool for cleaning up toxic chemicals released into the soil and water by industrial processes. Many prokaryotes are capable of degrading pollutants such as oil, solvents, and pesticides. In 2016, researchers discovered bacteria that can metabolize plastic There are two major branches of prokaryotic evolution: bacteria and archaea. Archaea are abundant in many habitats, including places where few other organisms can survive. The extreme thermophiles, or heat lovers, live in very hot water, sometimes near boiling. The extreme halophiles, or salt lovers, are found in areas like Utah's Great Salt Lake, the Dead Sea, and seawater evaporating ponds that are used to produce salt. A third group of archaea are the methanogens. Methanogens live in anaerobic or oxygen-free environments. They produce methane as a waste product. Methanogens are found in mud at the bottom of lakes and swamps and in solid waste landfills. Great numbers of methanogens also inhabit the digestive tract of animals. In humans, intestinal gas is largely the result of methanogen metabolism. Methanogens aid digestion in cattle, deer, and other animals that depend heavily on cellulose for their nutrition. Bacteria and other organisms that cause disease are called pathogens. Endotoxins are chemical components of the outer membrane of certain bacteria. All endotoxins include the same general symptoms: fever, aches, and a dangerous drop in blood pressure, which can lead to septic shock. Septic shock can kill a healthy person in a matter of days or even hours. Examples of endotoxin-producing bacteria include the species of Salmonella that causes food poisoning and typhoid fever. Staphylococcus aureus is the leading cause of food poisoning, and Neisseria meningitis is the bacteria that causes meningitis. One of the greatest threats is from endospores of the bacterium that causes anthrax. When anthrax endospores enter the lungs, they germinate, and the bacteria multiply, producing an endotoxin that eventually accumulates to lethal levels in the blood. In a terrorist incident in 2001, 5 people died when anthrax spores were mailed to members of the news media and to the U.S. Senate. Lyme disease is caused by a spirochete bacterium carried by ticks. Disease-carrying ticks live on deer and field mice, but they can also bite humans. Lyme disease usually starts as a red rash shaped like a bullseye around the tick bite. Antibiotics can cure the disease if it is administered within a month of exposure. However, if untreated, Lyme disease can cause debilitating arthritis, heart disease, and nervous system disorders. Because there is no vaccine, the best defense against Lyme disease is public education about avoiding tick bites and the importance of seeking treatment if a rash develops. Unlike other biological agents, the weapon of Clostridium botulinum is an exotoxin that produces botulinum. Botulinum is the deadliest poison on Earth. It blocks transmission of the nerve signals that cause muscle contraction. Botulinum results in paralysis of the muscles required for breathing. 30 grams of botulinum, which is a bit more than an ounce, is enough to kill every person in the United States. The term Protists is not a taxonomic category. Recent genetic and structural studies shattered the notion of Protists as a unified group. Some Protists are more closely related to fungi, plants, or animals than they are to each other. Protists is a bit of a catch-all category that includes all eukaryotes that are not fungi, animals, or plants. Most, but not all Protists are unicellular. However, all Protists are considered eukaryotic. Photosynthetic Protists belong to an informal category called Algae, which also includes cyanobacteria. Algae may be unicellular, colonial, or multicellular. Other Protists are heterotrophs that acquire food from other organisms. Some heterotrophic Protists eat bacteria or other Protists. Some are fungus-like and obtain organic molecules by absorption. Others are parasitic. A parasite derives its own nutrition from a living host, which is often harmed by the interaction. For example, the parasitic trypanosomes attack human red blood cells. These cause sleeping sickness, which is a debilitating disease common in 2 parts of Africa. Other protists are mixotrophs, which are capable of both photosynthesis and heterotrophy. Euglena is a common inhabitant of pond water. It can change its mode of nutrition depending on the availability of light and nutrients. Protists that live primarily by ingesting food are called protozoans. Protozoans thrive in all types of aquatic environments. Most species eat bacteria or other protozoans, but some can absorb nutrients dissolved in water. Protozoans that live as parasites in animals, though in the minority, cause some of the world's most harmful diseases. Flagellates are protozoans that move by means of 1 or more flagella. Most species are free-living and non-parasitic. However, this group also includes some harmful parasites that make people sick. One example is Giardia. Giardia is a common waterborne parasite that can cause severe diarrhea. Another flagellate is Trichomonas, which is a common sexually transmitted parasite. Trichomonas can also be contracted by ingesting food contaminated with feces that has the parasite. The parasite then travels through the reproductive tract by moving its flagella and undulating part of its membrane. In women, Trichomonas feed on white blood cells and bacteria living on the cells lining the vagina. Trichomonas also infects the cells lining the male reproductive tract, but limited availability of food results in a very small population size. Consequently, males typically do not have any symptoms of infection. Other flagellates live symbiotically in a relationship that benefits both partners. Termites are notoriously destructive to wooden structures, but they lack enzymes to digest the tough, complex cellulose molecules that wood has. Flagellates that reside in the termite's digestive tract break down the cellulose into simpler molecules, sharing the bounty with their hosts. Amoebas are characterized by great flexibility in their body shape and the absence of permanent organelles for locomotion. Most species move and feed by means of a pseudopodium, which is a temporary extension of the cell. One species of parasitic amoeba causes amoebic dysentery. This is a disease responsible for an estimated 100,000 deaths worldwide every year. Other protozoans with pseudopodia include the forams, which have shells. Although they are single-celled, the largest forams can grow to a diameter of several centimeters. 90% of forams that have been identified are fossils. Apicomplexians are all parasitic, and some cause serious human disease. They are named for a structure at their apex or tip that is specialized for penetrating host cells and tissues. Apicomplexans include Plasmodium and Toxoplasma. Plasmodium is the parasite that causes malaria. Toxoplasma gets transmitted through the handling of cat litter of outdoor cats. A woman who is newly infected with Toxoplasma during pregnancy can pass the parasite to her unborn child, who may suffer damage to the nervous system as a result of the infection. Ciliates are protozoans named for their hair-like structures called cilia. The cilia provide movement of the protist and sweep food into the protist's mouth. Nearly all cilia are free-living or non-parasitic, and include both heterotrophs and mixotrophs. A paramecium is a great example of a ciliate. Slime molds are multicellular protists related to amoebas. Although slime molds were once classified as a fungi, DNA analysis showed that they arose from different evolutionary lineages. Like many fungi, slime molds feed on dead plant materials. Two distinct types of slime molds have been identified: cellular slime molds and plasmodial slime molds. Cellular slime molds exist as separate, individual, amoeba-like cells for most of their life cycle. They only come together to form a mass when conditions require it, such as when food is scarce. Plasmodial slime molds form a large multinucleated mass of protoplasm, essentially acting as a single giant cell where many nuclei share a single cell membrane. Many unicellular algae are components of phytoplankton. Phytoplankton are the photosynthetic organisms that are mostly microscopic and drift near the surfaces of ponds, lakes, and oceans. Dinoflagellates are abundant in the vast aquatic pastures of phytoplankton. Each dinoflagellate species has a characteristic shape reinforced by external plates made up of cellulose. The beating of two flagella in perpendicular grooves produces a spinning motion. Population booms of dinoflagellates sometimes cause warm coastal waters to turn pinkish-orange in a phenomenon known as red tide. Toxins produced by some red tide dinoflagellates have caused massive fish kills and can be poisonous to humans as well. One group of dinoflagellates resides within the cells of reef-building corals. Without these algal partners, corals could not build and sustain the massive reefs that provide the food, living space, and shelter that support the splendid diversity of the reef community. Diatoms have glassy cell walls containing silica. They store their food reserves in the form of an oil that provides buoyancy. This buoyancy keeps the diatoms floating as plankton near the sunlit surface. The organic remains of diatoms that lived hundreds of millions of years ago are thought to be the main component of oil deposits. Green algae are named for their grass-green chloroplasts. Unicellular green algae flourish in most freshwater lakes and ponds, as well as many home pools and aquariums. Colonial forms of green algae include volvox. Each volvox colony is a hollow ball of flagellated cells. The balls within the volvox are daughter colonies that will be released when the parent colonies rupture. Seaweeds are classified into three groups, based partly on the types of pigments presented in their chloroplasts: green algae, red algae, and brown algae, some of which are known as kelp. Thank you for watching.